Right, shall we start? Cool. Okay. Welcome all back. This week I'm going to venture in the next sense, the second most interesting, arguably, sense, which is the vision, the visual sense, but I might be slightly biased here in my opinions. Talking about the eye and the brain, and in fact the fact that the eye is part of the brain. Again, as before, I want to concentrate first on the physiology, the anatomy a little bit, give you an overview of what's actually happening, and then trying to give you an indication of what processing lies behind. What processing lies behind what happens in the brain to make sense out of the confusing information. This will be a two-week part. So this week we'll talk about the basic perception in the visual world, that is mainly location and color, and receptive fields, and we'll do a little exercise, a practical later on, with receptive fields, which is not an experiment for yourself. It is an experiment. It's a simulated experiment where you need your laptops again. I hope you brought them. Okay, a little bit of physics to start off with. Vision is obviously the perception of electromagnetic wavelength. I hope that doesn't come as a surprise to anyone. It is just a very, very small spectrum of what the electromagnetic spectrum has to offer. So this is not the whole one, but it goes from 10 to the power of 12, what is it, in nanometer, to 10 to the power of -3, gamma rays, X-rays, ultraviolet. This is the visual part, infrared, heat, microwave, and it goes on and on and on. So we kept talking nanometers to micrometers to kilometers wavelength, and only this very small part, 700 to 400 nanometers, which is half a micrometer, so half a millionth of a meter wavelength, is what we perceive as visual light. Anybody know why this specific part of the spectrum? Why are we not sensitive to X-rays or, well, heat, for that? Yeah, an idea?
I believe it has something to do with the fact that our eyes are coated in water or something. So
yeah, that is true. It's an evolutionary reason, and the reason is that, as you say, underwater, there's only very small part of this is actually not absorbed. Actually, it is outside of the water. It's due to the atmosphere. It's outside of the water as well. Everything else gets absorbed in the atmosphere very, very quickly, or is not of interest of us because it actually isn't very much around. So there are no gamma rays around for us to make any sense of. There's no evolutionary purpose to do that. There is an evolutionary sense or purpose of being sensitive to heat. That could be very good if you're hunting in the dark and you want to see what's out there. You know, all the infrared cameras that can make that visible. And animals have, in fact, developed that. There are snakes that can see infrared light. Problem with that, it gets absorbed very, very quickly, and it doesn't get very far. And you also need to be quite cool to actually perceive it. But what we see is blue to yellow to red, the rainbow. I'm sure you've seen that. The color that you see here, is that a physical thing? Is that on the whiteboard, or is that on your head? Here it is again, the correlation between the outside world and the inside world. In the outside world, this is just a wavelength. There is nobody in the world that says that you should see 400 nanometers as blue. That's your brain telling you that is blue. And remember the very first lecture, is your blue you're the same as my blue? Who knows? There are indications for that, but this is not quite simple. But the world would probably look like the Matrix if we weren't able to put the colors to the perception of different wavelengths. It would look, you can imagine other ways of perceiving that. You could imagine in the auditory world we also have wavelengths that go from high to low, and we perceive them not as color, we perceive them as pitch. Very, very different. And here we in our head make some things to make us perceive this as wonderful, pretty colors, which is kind of nice of evolution if you think about it, because it could be other ways of perceiving that. Well, how does it happen actually in the eye? Here's a little bit about the optics. There's a picture of the eye. We've got pupil, the iris. The iris is the part that is colored, makes our eyes colorful. It's effectively part of the muscle that changes the dimensions of the iris, which then are focusing the incoming light onto the retina at the back. The eye is filled with vitreous humor, which is a egg-white matter, egg-white similar matter, very transparent obviously. And as you all have probably seen, there are things floating in that. If you look at the bright sky, bright blue sky, the high polarization index, you can see those very easily. It's completely normal. Don't worry about that. You can even see the blood vessels actually. If you do the right thing, you could, if you in a very dark night or dark room, put a little light against your eye, you can see the shadows of your blood vessels appearing in your visual field. What you see if you look through the pupil is the retina, and the retina itself, here it isn't shown, is actually part of the brain. It's the only part of the brain that you can directly see. It's in fact the only part in the human body where you can see blood vessels. That's why it's so interesting for doctors to look into your eyes, because they can get an idea about the functioning of your brain and the blood vessels from just looking into your eyes. The only way how you can see them. Okay, there are things that can go wrong. Namely, if the lens changes a form, or the shape of the whole of the eye changes, the focal point could be disrupted. Here, for example, hypertropia is when the eyeball becomes a little bit changes its pressure from inside, and so the focus point is behind the retina. But that can be all corrected usually with glasses, for example. But I'm not going into what's going wrong here. This is optics. I want to concentrate on the nervous side of things. But what's quite obvious is if we have this kind of projection, as you can see, this is the letter R looking at, it is projected the wrong way around and upside down. Yep. And it's projected here A, B, C, just exactly the opposite, A, B, C. So everything on the left side is projected to the right side of your retina, the right side is projected to the left side. Doesn't seem to hinder us seeing, obviously, though. Okay, there's another figure that another picture that concentrates on the tiny little bit of the fovea here. So light comes through the pupil and gets focused here on the fovea, and the fovea under the microscope reveals an interesting pattern, a set of structures of layers that from the beginning to the end is defined by very determined and very well defined cell types that we'll talk about in a little bit, which are ganglion cells, and then at the back the photoreceptors finally. I'm sure everybody has heard of the cones and the rod. We're going to repeat that a little bit, what's actually going on with these, why we have these, and what are they doing. So first thing maybe that I always find curious is that the photoreceptors, this is where the light is actually perceived. They're at the wrong end of the eye, aren't they? If you were to design the thing, you would, because the light has to actually go, well, it has to go obviously through the vitreous body, but this is transparent, but it then has to go through all of the neurons first to be picked up by the photoreceptors. Not every animal has that. Some animals, like octopus for example, have that the other way around, and it makes sense because they lose less light here. The issue though is, or there are some advantages of having it that way around, which is first of all that behind the photoreceptor is a pigment epithelium which is dark. If you look into your eye, actually what you see, apart from the red blood, is that it's actually dark. Do you know animals where that isn't the case? I'm sure you all know one. Cats. What happens if you shine a light into a cat eye? It's a cat eye. Haha. It reflects the light. This is where the cat's eye comes from. They actually have, rather than having a black piece of lining back here that we have, they have a mirror. So every piece of light that gets reflected in here gets that's refracted in here gets reflected the same way out. The problem with that, it's not the problem, there's actually a real advantage. Why it is an advantage for us to have it back, because we don't have this kind of mirror, is that we are less susceptible to scattering. There are two ways of looking at light. One way is looking at it in terms of wave, as you probably know, and the other way is looking at it in terms of photons. Both are correct, and both are actually needed to understand what's going on, because the refraction that's happening when light hits our lens is a feature of the wave theory of light, because it gets refracted onto a point of the retina. But what is actually then perceived is not the wave as such, but the photon that hits the photoreceptor. Photon receptor or photoreceptor, same word. But if it gets scattered back, if it doesn't get absorbed by one of our photoreceptors, it hits the wall and it gets scattered away. Let's imagine light comes from there, gets refracted over here, and gets scattered back there. That's bad. We don't want that. So it does have to be as dark as possible for us to have the least amount of scattering in there. The other reason is purely physiological, because this epithelium is also there to supply the cones and the rods with the energy, with the ATP that makes it then possible to have the chemical reaction to respond to light. Okay. Who here has heard of the blind spot? Who hasn't? Everybody. Good. What is the blind spot? That's the part where the optic nerve leaves the retina. This here, by the way, is the fovea. It's a little. I have a slide later where we see that in blown up. It looks a little bit different from the rest of the retina. Where all of the optic nerves go out, and the blood vessels as well, there are no light-sensitive receptors. So this is the spot where we don't see anything. We don't see it in our line of sight, and we'll have a little demonstration for you that you can do yourself to see if you actually see that, or what happens with what does your brain do to convince you that you're not blind in a certain spot, which is quite interesting. Okay. Now, the eye has been investigated for a long time. Actually, 200 years ago, guy René Descartes, a philosopher, already investigated in frogs. They seem to be very simple to dissect, and they have big eyes, and people knew about the connections, and they started thinking about what the connection between the retina, where the vision obviously happens, and the brain is. And our old friend Ramon Callao, who we met when we were describing neurons, because he drew all these fancy pictures of neurons beginning of the 20th century, he dissected frogs as well, and he came up with this crossing hypothesis, which is quite interesting. The problem is, this is the frog with these two eyes, and this is its visual field, and this is an arrow from one side to the other. You've got the two eyes, so the projection of this part of the arrow is in here, and this part of the arrow is in here. Now you've got the nerves, and if it was uncrossed like that, you would actually have a point in the brain where the arrow is the wrong way around, or it's just broken. Okay? So when you dissect a frog, you find that this is a much better representation of what's actually happening in the frog, namely that these two nerves are crossed. It's called a chasm, crossed, which enables the hemisphere, the retinotopy, the visual field, to be a complete connection or a complete representation of the outside field. It's the wrong way around, but that doesn't matter very much. And this satisfies exactly our retinotopy. You remember maybe the tonoptopy from last week? So that everything in the outside world that is close to each other, be it in frequency, be it in space, be it in vision, is represented in the brain close to each other. General principle. You remember the homunculus, where you have a representation of your whole body in a small, well, in a slice of your brain, and everything that is close together on my skin is close together on my brain. In the retinotopy, you want the same principle, namely, you want to have that. You don't want to have that kind of split. That at least was what Ramakayal suggested, and that was a good theory. Unfortunately, it turned out it's completely wrong. The eyes don't really care about, or the brain doesn't care that very much about that. And this is actually a representation of how it is represented much better in the frog. So there seems to be that kind of chasm. But in humans, this becomes a little bit more complex, because it is not. Humans do not have two separate visual fields, one in this direction, one in that direction. So you'll know your eyes are front facing. Our auditory visual fields both go forward. We'll see in a second what that results in. Okay, there's a human auditory pathway. Looks exactly slightly different. We've got the left visual field, right visual field. This is red and green, and you see that each eye has a red and a green part, so right and a left field. Now all the green ones are collected in the optic chiasm and go to the left visual field via the visual thalamus, and the red ones, so the left visual fields, go to the right side. Okay? Now this is the kind of the representation of what's happening in the visual neural pathway from sensory to cortex. If you maybe remember last week when we had a look at the auditory pathway, there were 7 synapses between the hair cell and the cortex. Here we have 1. That reflects a number of things. That reflects that a lot of processing is already going on up there. It also reflects that this system is a lot evolutionary younger than the auditory system, because it just didn't have time to be diversified, if you like, so much as the auditory system. It also means that the processing in the auditory system is actually much more involved. The simple reason for that is it's faster. It has to be much faster than the visual world. These are the superior colliculi that we already saw. They're the neighbors of the inferior colliculi, where all the hearing happens. So these are practically where the vision and the hearing interacts. And there's a sight line, if you like, that comes from synapses in what's called the pulvinar nucleus here. They go in the superior colliculus and they control the eye muscles. So this is the direct feedback loop from the eye through the superior colliculus back to the eye muscles. They're not drawn here. Why is that useful? It makes you able to stabilize your eye onto something. With something completely uncontrolled and unconscious, if you focus on something, you can move your head around as you like. You will always focus on the same spot. That's what your superior colliculus is doing for you. Right, back to René Descartes. That's an interesting story about how science worked 200 years ago. At the end of the 18th century, we wouldn't call that science today, it was philosophy effectively, but they did what they could and they had some interesting ideas. So he observed actually in the frogs that you have these 2 eyes in the frog, obviously, that you have the separation into left and to right eye, but you do not have 2 pictures in your brain, neither do you have 2 consciousness or 2 beings in your head. Although you have 2 brains, you've got the left half of the brain and the right half of the brain, but you do not have 2 ways of looking out of your eyes. You only have 1. So he was looking for structures in the brain that only exist once, and he only could come up with one, which is the pineal gland, which is a little gland a few millimeters which is hanging back from the auditory cortex, basically back to close to the hypothalamus, and he suggested that to be the place of the soul, because it's the only place in the brain where you only have one. So this needs to have both information from the left and from the right. Interesting step of faith, if you like. But yes, it is more interesting, more complicated obviously, but more interesting than he could imagine, because what really happens with this integration is highly complex and enables us to do a lot of things, like 3D vision, for example. Okay, let's have a look at our photoreceptors with an electron microscope, and we'll see there's a cutting through the retina. This is a look on top of the retina. You've got these rods and the cones. Here you can't see why they're called rods and cones. Here you can, maybe. These are the rods, look like rods if you like, long and thin, whereas the cones look more like, I don't know why they're called cones. They don't look like cones to me, but this is what they are. They're different from each other, that's the point. And if we look, if we draw them a little bit more schematically, here they look like a cone, funnily. Here they look like a rod, but in reality, obviously, they're not like that. The point is that the real difference between them is looking from the top. Is that surface area here is much bigger than for the cones, and that's obviously the trick, that the cones have a much bigger surface area here where it matters than these ones, because this is where the light comes in. Yeah, this is the light comes from top to bottom, but it has only the chance to hit something in here in this volume, and this volume is much bigger than that volume, partly because it's longer, but also partly because it's wider. Okay, human eye has about 10 million rods, 120 million cones. The structure of these look completely different from hair cells. Yeah?
So which one has the biggest surface area then?
Not the surface area, it's the volume that matters.
Oh, okay.
Because the volume is filled with rhodopsin effectively, you'll see in a second, and the more volume you have, the more chance that the cells have to catch a photon. So why are they similar to hair cells? Remember the hair cells in the ear? Because they are not nerve cells, they're sensory cells. Sensory cells mean they respond to something in their environment, in this case light, and as a result of that they change their membrane potential, and then we have these endings, but they don't have, they're called synaptic endings, but they do not have action potentials. So they generate a membrane potential, and this is picked up by secondary cells, bipolar cells in this case, that translate that chemical potential into an electrical potential. Got then these mitochondria for the housekeeping. They provide the energy, as we know. This is where the nuclei sit, and the signal goes from the bottom to the top, if you like. This is just very roughly what happens with one of these rods in the dark, in the light. Not going into any chemistry here. This is one of these rhodopsin. I find that a fascinating figure, because it goes to show this isn't the rhodopsin. This is the rhodopsin, but it changes the configuration of the sodium channel in here, and this looks like random ribbons, but it's actually a very well understood protein. So don't know what the colors mean, but we can write them down, the protein structure, in chemical terms, because we know how it is coded in the DNA. And the way that it is coded is one of the informations. The other way is how it's folded. So each of these foldings actually make a sense. What happens to the photon somewhere hits in here and leads to a structural change of the rhodopsin, and it splits into two parts. and that in itself then leads to, as the rhodopsin up here, leads to a release of a transmitter which closes the sodium channels. Now the logic here is unfortunately a little bit the wrong way around. Namely, in the dark, all of our sodium channels are open. This is called the dark current. So what I'm explaining in the next 10 minutes, 20 minutes, is all implying the other way around, namely light increases the activity. What it's really doing is actually decreasing the activity. But because this is so confusing, most people choose to explain it then the other way around and just change the signs, if you like. So we have a light comes in, our rhodopsin closes our sodium channels, sodium stays outside, and we increase the membrane potential, which leads to a reduction of spikes. But for the sense of understanding what's happening, we might as well say the light increases the activity of it, although the activity is then a reduction of spikes. But don't worry about that. Just remember, this is the way that it works in here. These are the rhodopsin molecules, and the structure is long and thin because we've got lots and lots and lots and lots of these rhodopsins. So we've got lots and lots of chances of hitting one of these with one of the photons. Now we've got rods and cones. We have three different kinds of cones that are usually called blue, green and red, and this is where our color perception comes from, because they have slightly different opsins. Rhodopsin, forgot what the other ones are called, and they're sensitive to different wavelengths. These are the blue ones, around 450, green 570, and red 620. And then we have the rods, which are slightly broader and have only one maximum. Okay. The fact that we have these three different kinds of cones is the single one fact why we see the colors that we do. Because we interpret the output of these different sensory units as being from a different type, and we make them into a perceptually continuous change of gradient of color. You've heard, you know, the color wheel, which is closed. If you put that into a color wheel, you put this end into that end, and it makes sense, because that then the color that is in between here and here physically doesn't exist, but perceptually does very well. I forgot what it's called. I think it's morph, which our brain makes up to give us the logic that this actually isn't a linear changing physical medium, but actually circular. Right, this is what was called the Trichromacy theory, because we've got our three main colors, primary colors, blue, red and green, which comes to the, well, blue, red and green, which is summarized in this theory of additive color. It's not a theory, well it is a theory, but it's so well used that it's well accepted now, that if you put these together you make up other colors. It's called additive because if you add red and blue light, you get magenta, blue and green gives you cyan and yellow, and if you add all three you get white. That only works with light though. You add the frequencies, you add energy of all of these colors, and that's why you add them together to get white. That's not what happens if you do it with colors, obviously. Yeah, you know the difference between additive and subtractive colors. In the subtractive color, it's called the CMYK, cyan, magenta, yellow, black theory model. The three primary colors are usually cyan, magenta, and yellow. And this is used for computer monitors, for example. If you zoom in on a computer monitor, you can actually zoom into here, if you like, with a magnifying glass. You would see that this magenta is actually made up of blue and red dots. just because you're sitting so far away you can't see the difference between them. But on a printer, computer printer, you have a different color wheel. Usually what are the colors if you have to refill the toner? They're not red and green and blue, but they're actually magenta, cyan and yellow, which gives you, for a printer, the reason is it gives you a greater spectrum of colors. You could do it with these three colors, but usually these are chosen, and black as well to give it more depth. So these are the three, always three colors we need to describe the world of our color perception, which comes from the fact that we have three different kinds of cones. Some animals have only two, like dogs for example. Some animals like birds have four. What happens if you only have two cones? You can imagine, because there are people out here, maybe even this room, who have only two types of cone, because that's what's called color blindness, or simple color blindness. There are a number of color blindnesses, but one of the most frequent one is where the green cones specifically do not work, and then people become red and green blind, but they can still see the difference between blue and yellow. What birds see is an interesting interesting question, because they see this is the result of having three. I can only imagine that this is something more three-dimensional. They see a completely different range. They must see colors that we have no concept of, because they can combine, instead of 3 dimensions, they can combine 4 dimensions of color. Yeah, but color and perception is not only based on what's happening on the retina, but it happens it's affected by what happens in your brain, and this is your previous experience, and also other things, context around. So for you, are these the same color, this one and that one? Looking at that? Probably not. If you overlay it with something, you see that they actually in fact are the same color, but your brain tells you that this one is darker than that one. Why? Because experience tells us that the sun is in the sky and shines on the top of an object, which, if it is that grey, must be darker than it is at the bottom in the shadow. So color is not only affected by cones and rods, but by experience like that as well. And I don't know if you've seen that picture recently. I don't know what people think about that, but is it now? Make an experiment here. Who sees it as black and blue? Me too. Who sees it as white and gold? It's 50-50. Unbelievable. Do you know why? No, nobody knows. No, people do know why, but why? After what I just explained, why do people split right through the middle on the perception of it?
The way you put it in perspective, so if I were to view that as like a dress in incandescent lighting,
Yeah.
the brain sort of then
Exactly.
corrects the colours in a sense.
Exactly.
If I were to perhaps cut out the background and consider that I was looking at it in like a digitally.
I was trying to do that. Anybody changes their mind in terms of what it was?
It was something to do with like the different images you see it on, because like I showed my friend on her phone, as you see a different image on my phone. So
Yeah.
it's to do with like the pixels and the light that you're perceiving it. Like the light in here is similar. So we
But it's the same pixels that everybody perceives, but your brain perceives something different on the same physical pixels. So no, I don't think that is something to do with the actual picture. It's something to do with your experience and the environment around it. That's why I was trying. It's a little bit experiment of mine to see if somebody changes their mind. Because if you see it in this blue background, it is a different lightning. It's obviously a question of lightning. It's exactly the same phenomenon as that. You see, you expect the sun coming from the top. That's why the top one must be darker. So here, it depends if you see that, if you think this is in a bright environment or if it's in a dark environment.
Is there a reason why it would be white and gold though? Like so far away from blue and black?
I don't know. I really
You
don't
said
know.
it, and when I went up in the morning in the set light, it looked white and gold, and then I went down
Yeah.
to seeing it looked black.
Yeah, that's the context, exactly. That's because you look at it in a different light, in a different and maybe brighter room, and maybe different background setting. That's what I was trying to see. I think if we did that experiment, you close your eye and you didn't make up your mind, you might have chosen differently in that kind of background. But that's exactly the point that I'm trying to make. The color is in your brain. It's purely in your brain. It's not really out there in the real world.
What colour, if you were to completely isolate the pixels,
Yeah.
what sort of shade would they be?
Oh, it's blue, gold and white.
It's gone white.
Yeah. If you Google color dress or something, you find good explanations.
It's blue and black.
Blue and black? Oh.
Black, but the pixels themselves, the colour of the pixels, if you were to isolate them individually.
Yeah, yeah.
Oh, well, on that photo?
Yeah.
Yeah.
No, they do have.
They have the black on Photoshop, but if you...
Oh, really? Okay, I misunderstood then.
I mean, yeah, you can Photoshop it. It's exactly like here. You can Photoshop it in a way to make it more obvious, and then every pixel has a defined physical color, but you don't register that.
I've always seen it golden.
Yeah, so like, I can't make it go
And
black.
when I watched the news article.
That was on the news.
And it had them go to the actual shop.
Yeah.
And they got the dress out, and it was blue and black.
Yeah.
And I was like, obviously it's blue and
Yeah.
black. But on the photo.
On the photo is dangerous because it doesn't show much of the background. It doesn't show the context in which it is, in which you would see it on the television or in real life, obviously. In real life, we're very well suited to see something as continuous, independent of the light surrounding. You can you can switch the light here off almost, or you can make it really bright. You would almost always see these seats as blue, because your brain makes a correction for that, and it cannot make this correction because it hasn't got enough information from outside to do that. Yeah.
I was going to say, when they inverted the image, if you saw blue and black, you could kind of see how it went white and gold, but you saw, so most people see the blue bit as white and the lace as gold.
Yeah.
If you inverted it, you saw the dress as gold and the lace as white.
Okay. No, that doesn't make No, it's about context, that's my point. Cameras, by the way, you have a feature on a camera which is called a white correction, which you can do on a more expensive camera. So you tell the camera this is white, for example, by taking a picture of a white piece of paper. And then the camera knows this is physically white, and it will correct everything else according to what you told it was white. The problem is that this white changes with the light. Because if it's darker, the white isn't white anymore, it's grey, or whatever the equivalent in physics is. Okay? So this is what's going on here. So I'm disappointed that it doesn't work. I wanted to give it a different context, because your eye now would be actually focusing more on the context of the blue, and that would enhance the colours. Anyway, it doesn't work. Sorry.
So has it got more to do with our previous experience?
Yes.
Because right now we're all seeing it in
Yes.
context.
Yes. Yes, and that it splits right 50-50 shows that everybody has a very different experience in that matter as well, which is super interesting. So this, I have not seen such a clear case of where really half of the people see it completely different. But this is the actual, for me, one of the most fascinating aspects, which we really discover from hearing, the range of vision, of luminance. This is a CD is a candle. It's candela, I'm not quite sure. Candle. This is one candle means one candle, literally one candle 1 meter away. Okay? Imagine this room lit up with one candle. That wouldn't be very bright, would it? But it's still actually quite bright in comparison to what you are able to see. Because you technically see a candle from, what is it, half a mile away or something, if you would do that experiment in a very quiet night. But the range goes from 10 to the power of -6 to 10 to the power of 6. This is 100,000? No, hang on. There's 1,000? 1 million candles 1 meter away from you. That's a lot of candles, isn't it? But that's bright sunshine, obviously. You wouldn't want to be looking into the sun. You actually can get blind from looking into the sun because that's how bright the sun is. But this is the range of a candle half a mile away to 1 million candles, and you still see the colors continuous. You see the same colors because your brain accumulates adapts to that fantastic range of brightnesses. Very similar to the 12 orders of magnitude of sounds. If somebody speaks very quietly or speaks very loud, still the same message. Your brain doesn't change the message because of the amplitude. It doesn't change the message because of the lightning. So the only thing here is the very dark ones perceived by the rods and the very bright ones perceived by the cones. And that has an effect on vision. Namely, the cones are the ones that have red, green and blue, and the rods have only one color, and that's why the nights all cats are grey. You can only see one color when it's not very bright. There's another issue about the cones and the rods, that they're distributed very unequally throughout the retina. There's a distance from the foveal center, the 0, and the red line here is the density of the cones. So this is the fovea in the middle. It's a very, very small area. They have a very high concentration of very small cells. You don't want many big rods in there. Why? Because they're big. They are more light sensitive, but instead of having one of these big rods, you can have 20 cones, which gives you a higher sensitivity. That's why we can see so clearly in our fovea and not outside. The sum of these two is pretty much the same. It doesn't change that much across the whole optical field, but you obviously have more cones in the middle and more rods on the outside. So if the further you go away, the more of the big blobs you have. So both have different advantages. In the middle, they're good for color vision and very sensitive, well, very small angle, and outside they're good for night vision, but bigger field. Okay, here's a little picture of the fovea to see what's going on here. The fovea basically remove all of our cells out of the way so that the light can directly penetrate our cones, which are very, very, very neatly arranged and directly exposed to the light. So all of the axons go to the sides, to the top and the bottom of those. Okay, so this is where fovea obviously were the most sensitive, highest density of the receptors. Right, talking about the retinal wiring, we've got our photoreceptors now here on top. We've got our horizontal cells, bipolar cells, amacrine cells, and our retinal ganglia. This goes to the brain. Now we've got, what did I say, about 120 million of these photoreceptors, but we only have about 10 million retinal ganglion cells. So if you say you have 100 million, let's say that was split up into 10 million rods in the fovea specifically. So you've got about a 10 megapixel camera in your eye. But the information that actually goes to the brain is reduced a lot. So the eye itself, with this setup, does a lot of information reduction. And it does processing with the vision to take out, well, to throw out 90% of the information and to keep the 10% that is important. And the 10% that we'll find out about at the moment is color and orientation. These are the two, well, actually it's not orientation, it's grading that we'll discover is the most important one. So this is why the retina is called part of the brain, because this is all brain processing. Okay, very schematically what's happening. These are the photoreceptors, and this is what I'm trying to explain now, is a receptive field. And these receptive fields always look circular. You're just looking at it from the side. This is why it's such an oval, with the pluses, meaning they're excitatory and the minuses they're inhibitory. So these photoreceptors, all of them project onto these horizontal cells, which go the top at the bottom, and in the middle you've got this bipolar cell. And the horizontal cells take out information from the photoreceptors and also to the bipolar cell, and that goes to the retinal ganglion cell. This is all that I want to understand. There are lots of other things going on, especially with the acromion cells, which are simply not understood well enough at the moment. But this principle, which is described here, of these receptive fields, is genius, and is very simple, and it's actually used everywhere. It's not only in the eye, it's in the ear as well, it's in touch, and so on and so on. And I'll show you how that is connected. First of all, we need this concept of the receptive field. This is how it has been developed. 1950s, people started sticking electrodes into the optic nerve, and then they put a little light on the screen, and if it's here, nothing happens. Here, nothing happens. If they put it there, the neuron likes it. People called that the receptive field. That's the area where light does lead to an excitation. So far so good. The receptive field of auditory neurons we saw last week. This is where a sound produces an activity. Here we have an area, it's an actual physical space. Last week it was a frequency, now it is a space where there is where something happens. And we have big and small and large of these, because we have cells, bipolar cells, and these ganglion cells, that either only connect to one of these or to many of these. This specifically happens in the fovea, because then we've got one of these ganglion cells connected to one of these cones, which means this is very, very specific, only one tiny part of your visual field. Whereas these ones can be very large. Why is that an advantage? Because it collects more light. Because they all basically act together to excite our ganglion cell, so they can maybe pick up a millionth of the light intensity than that single one. So that one only works in broad daylight, that one works in the night as well. Both is good, and we need both. In fact, we need a combination of both. And they're connected via this concept of the receptive fields. Namely, every neuron has a receptive field, which can be described by pluses and minuses, and they mean there's pluses and minuses. If you put a light on here, this yellow dot is always a piece of light. Hello.
Sorry.
You put a piece of they shine a little light in there, and if it hits the plus, you get an increase. So if nothing happens, they have spontaneous activity, these neurons, so you get maybe 10 spikes per second. If you hit the center, so the plus area, you get a lot of increase, and then it goes back if you switch it off. Or you put the light into this negative area, spot surround it's called, you get an inhibition, so you get 0 in that area. So you get this, first of all, this on center, they're called on center because sensitive to light here and switched off by those. This is behind that is a very, very general principle. It's called lateral inhibition. Inhibition because it suppresses, lateral because it comes from the side. Lateral inhibition is a principle that we had briefly in hearing, but it's actually a fact of every sensory. It's one of the main principles of brain processing. Here's an example from touch. If you touch a piece of your skin, then all the neurons in this area will notice it. But some of this is a little way of describing that. Some of these will notice it more, and some of these will notice it less. Now the ones from the side will inhibit the ones in the middle. Well, they all interact with each other inhibitly. They all inhibit each other. But the effect is that you get an area of sensation which is much smaller than the area of the reception. So lateral inhibition effectively squeezes the perception, makes it sharper. And sharpness is good because it allows us a more sensitive way of detecting, well, in this case, where the touch was, or in the visual case, where the light came from, or in the hearing case, which frequency was actually excited. And yeah, that's your task. How many black dots are there? That's the visual result of this lateral inhibition. Because what happens if you focus on one, there's a lot to see actually. If you focus on that one, you see it as white, I hope, yeah? Or black and gold, depending on the. But in the peripheral areas, you get a combination of blacks and whites, and there's an overcompensation because of the lateral inhibition, which makes the white dots appear black. This is a direct consequence of the lateral inhibition that overcompensates the whiteness in here. It's an artificial example. It's not something that you see in the real world, but if you do it like that, it's a very effective way of demonstrating that. Yes. So we have the on center. We also have the off centers, and the off centers work the other way around. Then you've got a negative in the middle, and then you have an excitatory field outside. If you put your spotlight in the middle, you get a reduction. If you put a spotlight in the outside, you get an increase. Now, why do I tell you all of that? Why is that interesting? Why don't we just need our cones one-to-one connected, centered to the brain? Well, first of all, information reduction, because we reduce our 110 million cones and rods to something like 10 million nerve fibers. So that's good. But we also do something more useful than that, namely we pre-process. We enhance a specific kind of information. We enhance something that perceptually is important for us. Now in the vision area, what is the most important thing, what you can imagine? Looking around in this room, what's the most dominant character of your visual perception? Ignore color. Color is one of them. The other thing, which one you couldn't live without? Detection of, it says there.
Contrast.
Yeah, contrast of what? Of edges. It's all about edge detection, which is contrast between two things. But everything in the world has edges. Well, probably not, but this is the way that our brain evolved to see the world. Because what we always concentrate on is edges. And it seems to be, I don't think this is a general law, but it seems to be the most efficient way to understand a scene if you concentrate on the edges of it. And what does this receptive field have to do with edges? Well, they do. They use the lateral inhibition. Lateral inhibition means plus in the middle, minus outside. Because if you put a stimulus on that, actually, let me talk. Let me use the next to explain it better. What it does is these receptive fields. That was the last slide. They can have different sizes. They can be small, medium. They can be quite large. But depending on what frequency, and this is spatial frequency, so there's a gradient in here. Depending on which gradient you have, and this gradient is an edge, effectively, you can detect either with the small receptive fields. You can you detect best these high frequencies. Medium detect best the medium frequencies, and the big ones detect the low frequencies. And that together allows us. Here's an example. Here's a picture of an American scent. Looking through large receptive field and looking at through a small receptive field, this enhances the edges. That's what I mean. This blurs out the edges and tells us something about the bigger picture, if you like, the brightnesses. So the brightness, the contrast here is much higher between black and white than in here. But here we see all the edges. So where there is an edge, every time there is an edge in here, you get a black line. This is just a consequence of these receptive fields, and that's why it's so useful. So rather than sending the visual information as we see it as the screen of the retina into the brain, what the brain actually sees is these edges mostly. And because we have different receptive fields, they can be of different frequencies, so different sizes of our visual objects. Okay. I think there was enough to say. I'll repeat that next week. Talking about eye muscles. I want you to make a 5-minute break and do our little experiment then. And first of all, find out about your blind spot yourself. I printed out that little experiment. I'm sure most of you have seen a blind spot at some point. Let me just pass that on. Get together in your groups, please. Make sure that you have a laptop. Go to this web page. What you see is that little experiment, hopefully, if everything goes alright. It has an introduction and a little bit of explanation in here, because I appreciate that you didn't quite catch everything with the receptive fields, but this is our receptive field, and I can do the experiment of putting our light straight into the receptive field or at the negatives, and I can see here, up here, what happens to the firing rate. And your job is to, well, change the spot size, large, medium, small. You can look at different receptive fields, and if you're confident with that, you can do a quiz where you now have to find out which of these types, if you're doing the experiment, which of these types you actually have, just based on your receptive field and the firing rate. And this is not simple, it's not trivial, because you actually have to have intuitive understanding of the receptive field. So I guess it will take you a few minutes to actually play around with that. And when you manage to find out which cell that is, I just guess if it's that one. No, it was the wrong one. It was actually that one. Okay, so your job is to be experimenter, do electrophysiological experiment, and to count firing rates. I help you with that as well. Yeah.
Does that mean in the surround field, if say it was the excitatory surround field and in the intracenter,
Yeah.
if the light covers the whole field?
So excitatory in the outside, like that.
Yeah.
Yeah.
If the light covers the entire.
Like this.
Yeah. Then would it equalise, or are there more positives?
No, it would equalize. Well, in theory here, at least they're exactly the same negatives and as positives. Yes, but find that out for yourself. It allows you to play with that.
It's all kind of like substantive anyway, isn't it? Like model kind of thing.
Well, yeah,
It is a model and it is a little bit simplified, but it's not too far away. Why are these things round? Here's a philosophical question for you. Why are they not oblong or upwards or diagonal? I just don't exactly know the right answer for that, but I can imagine. Why?